Hi guys, welcome to Unit 7, Lesson 2, Pmat. So in the last lesson we talked about what is mitosis, so that is cellular division, how cells reproduce. And today we're going to specifically talk about what happens in each of these steps in prophase, metaphase, anaphase, and telophase, also known as Pmat, just so it's easy to remember. First things first, we have a warm-up question. The warm-up says, DNA replication is called blank blank because half of the new DNA molecule is old and the other half of the DNA molecule is new. Good job. DNA replication is called semi-conservative because half of the DNA is new, so it's half of the DNA is old, so it's semi-conservative, but the other half of it is new. And this is what it looks like, right? If our original template strand, just for demonstration purposes, is blue, then as we're making a newly synthesized DNA molecule, the two sides of it unzip, right? And then using base pairing rules, DNA polymerase is going to add in those extra bases. That is how we create new DNA strand, and this is known as semi-conservative. Okay. Today's lesson objective is that students should be able to describe the process of mitosis and order the different steps. So hopefully you remember this cycle that we have been talking about. G1, the first growth phase, the S phase for synthesis of DNA, G2, which is the second growth phase, and then today primarily we're going to focus on what this mitosis actually is. So one misconception students usually have is that they assume that the way that you grow from a small baby into who you are today, so from a baby that is 5 days old to someone who is 15 years old, right, your body gets much bigger. And a lot of students think that, that just means the cells got bigger. No. Living things grow by increasing the number of cells. So they increase by doing mitosis. Their size cannot increase. And a lot of students are like, well, why can't the cells just get bigger? Well, big cells cannot survive because the larger that a cell gets, the more demands it places on its DNA, and it has difficulty maintaining homeostasis. Remember that word, homeostasis? Well, if you don't remember, homeostasis is that balance. Right? So a really big cell is going to have to spend a lot of energy and a lot of time removing waste and transporting materials around the cell. And that is honestly a waste of energy when cells could instead just divide up the work by dividing themselves and working together as a team. And that's how it works. So big cells cannot survive. What happens is that a cell is going to divide into two cells. And the way I remember this is that, you guys have all heard the saying, 2 heads is better than 1. Well, 2 cells is going to be better than 1, right? 2 cells can work together. So cell division. What is cell division? So before a cell gets too big, it has to divide. So one parent cell, remember that term parent cell, makes 2 identical daughter cells in the process of mitosis. So this is still a recap. So why does the cell cycle happen? Well, the cell cycle is going to serve 3 major purposes: for growth, for repair, and for reproduction. So let's look at that. So growth is going to be a purpose of cellular division, like we just said, from when you are born as a baby all the way up till what you are today, which is an adolescent. Your cells are multiplying, right? Your cells are not getting bigger. You're just getting more cells that have more specific jobs that can do more things. Bigger organisms have more cells. And how do they get more cells? Through the process of mitosis. Repair. So for example, all of us have gotten a skinned knee or a cut on their finger or something like that, right? How does that fix itself? Well, mitosis. Mitosis replaces those broken, those damaged, those dying cells with new, fresh cells that have the same abilities. And of course, reproduction is a purpose of cellular division. But one thing to note is that reproduction is a different process than mitosis. Okay? Again, listen to me. Reproduction involves a different process than mitosis. It involves meiosis, which is MEIOSIS. Okay, and we'll have a separate lesson on that later on this week. Okay, so just so you know, It is still a type of cell division. There are two types, mitosis and meiosis. So we'll talk about meiosis later, but today we're focusing on mitosis. Okay, so remember the three phases of the cell cycle. Hopefully everyone remembers this. We have interphase first. Interphase is where there is normal growth and DNA replication. Quick question, when does DNA replication occur? Did it occur in growth 1, S phase, or G2? Right, DNA replication occurred in S phase, the synthesis phase. That's what the S stands for, is synthesis. Good job. Mitosis. Mitosis is the part that we're going to focus on today, and that's going to be division of the nucleus. And of course, if you divide a nucleus, what's inside of the nucleus? DNA. So prophase, metaphase, anaphase, and telophase, we're going to go in depth about those. And of course, cytokinesis, which is just that final stage in which the cytoplasm, aka the jelly inside of the cell, is going to divide, and then you'll end up with two separate cells. Okay. Perfect. So interphase. This is another misconception that students have, is students think interphase is going to be quick. No, mitosis is going to be quicker than interphase. Interphase, most cells are going to stay in interphase for most of their life. Okay, so interphase is the longest phase of the cell cycle. All right, let's break it down again. So we have G1, the first growth phase. That is where cells grow. They're going to synthesize whatever proteins they need, and they're going to do normal activities. Remember that enzymes are a special type of protein. So DNA polymerase, DNA helicase. This one you don't have to know, topoisomerase. These are all different enzymes, which are proteins that must be synthesized before the cell can do cellular division, because they have to use those proteins to do their job. Okay, so that's here, that cellular growth phase. The next phase is going to be, a G1 checkpoint, right? Because our next phase, as you can see, is where DNA replication is. But before the cell can go out of G1 and into G2, there's a checkpoint. That checkpoint is basically where the cell is looking at itself, and it's like, is DNA actually ready for replication? Is it ready? If it's not ready, the checkpoint will not let the cell cycle continue. If it is ready, the cell cycle will continue. Okay, and once the cell cycle continues past the checkpoint, then we have S phase, which is known as the synthesis phase, where DNA replication is occurring. Okay, what's after S phase? We're going to go to G2. Before that, let's review DNA replication, because that also is an important topic that we talked about in the last lesson. So DNA replication is, again, this word semi-conservative. Well, what does semi-conservative mean? Remember, first things first is we have a DNA double helix, but it's going to unwind into a ladder shape. Where's my DNA at? Right. So this is known as the DNA double helix when it's in this twisted up shape. Just going to unwind whenever it needs to replicate. So this is the ladder form of DNA. Notice there's no changes. All it did was it was twisted up, and now it's not twisted up. So it has easier access for the enzymes. Okay. And then basically what's going to happen, it doesn't want to untwist. What's going to happen is the enzyme helicase is going to unzip. Okay. How does it unzip? Well, it's going to break a special type of bond. What type of bond holds the two strands of DNA together? Good job for reading or remembering. The hydrogen bonds are what are in between it. So the hydrogen bonds are going to break, which is going to separate them into two different strands, which I can't take this apart, but whatever. Okay. Once they're in their two different strands, then this thing is called DNA polymerase, aka what makes an enzyme that polymerizes, which we know a polymer is just a large molecule. It's just going to start adding in these bases using our base pairing rules. Okay. In the end, we have half of our molecule that is original and half of it that is newly synthesized. That is what is known as semi-conservative. Semi means half, right? So we have half of it is conserved and half of it is new, so it's semi-conservative. Okay. One thing to also note is that since we're using complementary base pairing rules, when we pull these two things apart and then we put in their other pairs, this is identical to the original. Okay, so that's how DNA replication occurs. Okay, now I have a problem for you. I want you to look at this DNA model of DNA replication, and DNA polymerase went in and added these bases. However, there is an error somewhere. I want you to tell me where that error is. Is it at position 1, position 2, or any of these? Tell me, where is the error? Good job. The error is definitely here. DNA polymerase made an error here because it put a T. Can T and T pair up with each other? No. Remember, our base pairing rules are cars in the garage and apples on trees. Okay, they always pair up that way with each other. Perfect. Okay, so you will learn this mostly in either AP Bio if you choose to do AP Bio as a senior, or in college if you do biology. You'll learn that DNA polymerase is actually an error-prone molecule, and that there are it's an error-prone enzyme, and it makes lots of mistakes all the time, which is why we have a checkpoint that we'll talk about in a little bit. Okay, so again, this is yet another model of DNA replication. The original DNA strand, they get pulled apart, right? Those bonds in the middle get broken. What were those bonds called again? Hydrogen bonds. Good job, good job, good job. Hydrogen bonds get broken. Once they're broken, DNA polymerase is going to base pair using our complementary base pairing rules. So in the end, we end up with two identical molecules that are semi-conserved, which means half of it is old and half of it is new. Perfect. Good job. Okay, then we have what is known as, so that was all occurring right here in S phase. Notice again the length of these phases. G1 looks pretty long. S phase is a little bit longer because it has to replicate all of the DNA in the cell, which is not an easy job. Then there's growth 2 phase, which is a little bit shorter, but notice that interphase is made up of G1S and G2, and that is the majority of the cell cycle. Okay, remember that interphase is the majority of the cell cycle. So within the second growth phase, the cell is just going to continue to grow and prepare for mitosis. It's just going to continue preparing for mitosis. But there's a G2 checkpoint. Before we can go into mitosis, before division can actually begin to occur, we have to check for something. Did DNA replicate correctly? Is the cell fully ready for division? If DNA is damaged, the cell will not continue past this checkpoint and on and into mitosis. Can you think of why? Like, why must DNA be repaired, be checked and repaired before the cell can fully divide? Well, of course, DNA must be repaired before cellular division so that each of the daughter cells ends up with the correct amount of DNA, right? Imagine that you have, I have nothing to model this with. Here, we'll do this. Okay, so imagine that you have a cell, and you, okay, no, that's a dumb analogy. We'll look at it in some other ways. So, mitosis is division of the nucleus. Okay, so what we just talked about was called interphase, which that was that preparation, that was a review from the last lesson. G1S, G2. Now we're moving on to the second part of the cell cycle, which is mitosis, and then we'll finish off with cytokinesis. So before we get into what mitosis is, there's some things that I want to clarify to you, because I remember when I first took biology, I was so confused on this, and I was like, I don't get this, so I'm going to explain it. So a spindle fiber is these little line things that you'll see in every picture of mitosis. Okay, these little line things. They're called spindle fibers. And basically what they do is they grab on, let's say this is a chromosome. What they do is they're going to grab on. So there's going to be, this is one side of the cell. Oh, this is perfect because this is a circle. Okay, so imagine this is DNA in the middle of the cell. The spindle fiber is going to grab on here, and another one is going to grab on here. They're going to pull half the chromosome. They're going to pull the other half of the chromosome away. Okay, so that each of the daughter cells is going to end up with half and half of the DNA. Okay, so that is what a spindle fiber is. Okay, and this term centriole is just what produces the spindle fibers. Okay, so again, the spindle fibers are what grabs half the chromosome and it's matching half of the chromosome. They both get pulled over into the new daughter cells, and the centriole is what produces the spindle fibers. Okay. One thing to note about a chromosome. So technically, this is not the correct model to use for this, but it's just because it's what I have. But basically, a chromosome is going to have multiple pieces of DNA into it, and basically, okay. So this whole thing is called a chromosome, but this is also called a chromosome, and that is also called a chromosome. These are also known as sister chromatids. They're sister chromatids. They're sisters because they are identical. Okay? They are identical. So a chromosome is just a condensed form of DNA where this half of it gets pulled over into this daughter cell, while this half of the DNA gets pulled over into that daughter cell. And what do they get pulled by again? What do chromosomes get pulled apart by? Spindle fibers. Okay? All right. If it doesn't make sense, don't worry. Like I said, we always do some practice with it. We're going to look at it in some different ways. So we have prophase. Prophase is a part of mitosis in which the chromosomes form, the spindles begin to form, and the nuclear envelope, aka what is surrounding the chromosomes, is going to begin to break down. It's going to disintegrate. Okay, prophase. Prophase. The first part of Pmat is prophase. Next up, we have metaphase. Metaphase. Chromosomes line up in the middle, and the chromosomes, each half gets attached to a spindle fiber. Okay, see that? This is one chromosome. One half of it is being pulled by that spindle fiber, and the other half of it is being pulled by this spindle fiber, and that happens to all the chromosomes. Okay, so that in the end, when the spindle fibers begin to pull, that half of the chromosome is going to go over here to this new daughter cell that's developing, and this half of the chromosome will come over here to this daughter cell. Okay, metaphase. The way that I remember what happens in metaphase is that chromosomes line up in the middle. Metaphase is M, middle is M. They line up in the middle. Okay, then we have what's called an M checkpoint. The M checkpoint is important because it basically says, okay, wait, is every single chromosome in this cell attached to a spindle fiber that is going to pull it equally, pull half and half to the new daughter cells? If they are not attached to a spindle fiber, will the cell continue on past this checkpoint? No. The whole purpose of a checkpoint is to check that everything is okay. If everything is not okay, the cell will not continue on. Right, so the M checkpoint, as with all of our checkpoints, we have the G1 checkpoint, we have the G2 checkpoint, and now we have an M checkpoint. Every checkpoint is basically designed to say, wait, are we ready for the next step? If the cell is not ready for the next step, it will not continue on. And that makes sense because you can't, cells make mistakes, so they got to double check their work. Okay, after prophase and then metaphase, next up we have the A, which is anaphase. Anaphase's job, or anaphase, is characterized by the chromosomes getting pulled apart. The sister chromatids, which are half of the chromosome, get pulled apart. Okay, how do I remember that? Anaphase means apart. Honestly, y'all should really be listening to me because when I tell y'all, my biology teacher didn't teach us this. I was like, I don't understand this. I remember these words that I'm telling you. Metaphase, middle. Anaphase, apart. You will never get it wrong. And then, of course, we have telophase, the end of Pmat, which is when two new nuclear envelopes begin to form around the newly separated DNA. Okay? Two new nuclear. So nuclear envelope just means like the beginning stage of a nucleus, obviously. Two, telophase. Makes sense. Just saying. Okay, now let's look at it all together. Pmat. Remember, these are the stages of mitosis. So first things first is we have interphase. What are the three phases of interphase? G1, then S, and then G2. Okay, after interphase is done, the cell is going to go into mitosis, which mitosis stages are prophase, metaphase, anaphase, and then telophase, which it's okay if you don't remember those words because I'm gonna be honest, most people don't. Just remember Pmat. It's much easier to remember. Okay, so we have prophase, which is at the very beginning. The cell looks like this. It's when the cell is preparing for division. So it's preparing for division by disintegrating the nucleus so that the chromosomes and stuff can get pulled apart. Then we have metaphase. Metaphase is when all the chromosomes are lined up in the middle. Then we have anaphase, where the sister chromatids, so half the chromosome is being pulled and the other half of the chromosome is being pulled the other way. They're being pulled apart. And then telophase is whenever we start to have two nucleus develop. Okay. If you're not writing this down, you should probably write this down. I can't wink. Okay, and then last but not least, we have cytokinesis, which is the division of the cytoplasm. So cytokinesis is when just the two identical daughter cells are being produced, so they are being split apart completely. So this is probably like the beginning of cytokinesis, and then they'll be and they have what's called a cleavage furrow. Cleavage furrow just is basically where it gets cleaved, which just means it gets chopped. All right. So now what? Now what does a cell do? It's gone through all this. It did G1, S, G2, did prophase, metaphase, anaphase, telophase. Now what? Well, now the cell's just going to return to interphase. Will continue normal growth and normal function until it's ready to divide again. All right, let's talk about cancer. So, everyone kind of knows what cancer is, right? We all know that probably all of us have someone, a friend, a family, or maybe even a teacher that you've known who's had cancer. And a lot of us kind of know like it's a disease, but we don't know exactly what it is. Well, I'll tell you, cancer is literally just uncontrollable cellular division. It happens when the cells just enter the cell cycle repeatedly, even when they don't need to. Okay? So, did you know any mutation that causes the cell to divide uncontrollably is known as cancer? When cells cannot stop dividing, that is cancer. Okay, that is literally the definition of cancer. When cells cannot stop dividing. In some cases, but not all cases, a tumor is going to form. What is a tumor? A tumor is just a big bundle of cells. But notice, not every cancer forms a tumor. Right? Some cancers, the cells will just divide, and they'll just like disperse throughout the body. So it's not really a tumor. It's just the cells are just all around the body. So in some cases, a tumor may form. But cancer is not characterized by tumors. A lot of cancers do not have tumors. Okay? What defines cancer is when cells are uncontrollably dividing. Okay, this is game Mitosis Mover. I'm going to go ahead and link this game so that you guys can play it if you want to review mitosis a little bit more in depth. Okay, so questions. I have a question for you. When would a cell do mitosis? Well, a cell's going to do mitosis whenever it needs an identical copy of a cell. So that can, happen in many different ways. So for example, when an organism needs to grow larger, so from a baby into a 15-year-old person who's watching this video right now, right? If you're growing larger, your cells cannot just get bigger. They have a max size that they can be. So instead of, cells growing really, really big, they're just going to divide and they're going to make more cells. They're going to work together for a common job, aka that's called an organ, by the way. We already know that. For example, if a red blood cell reaches the end of its 120-day life cycle, they only live 120 days. After that, they're too old, they're too damaged, so they have to divide into brand new cells. Or if you get a sunburn and you have a bunch of skin that's like blistering and falling off and crumbling and whatnot, then cells are going to need to be replaced, so they're going to divide and make identical cells. Okay. Another thing that's interesting about genetics and whatnot, how this is relevant to life, I think all of us probably know or have seen someone who has Down syndrome, aka trisomy 21, How does that happen? Well, I'm just going to show you. We're going to talk about genetic disorders a little bit later, but I just want to give you kind of an idea because whatever. How does Down syndrome occur? Well, if chromosomes are misaligned, so the spindle fibers didn't grab them correctly, and the cell continued on dividing anyways, somehow, because sometimes, like, it has a checkpoint, but does that always work? Not always. Sometimes, like I said, cells make mistakes just like anything else in the world makes mistakes. So if the chromosomes are misaligned and the cells divided anyway, then some of the daughter cells may have too many chromosomes and others may have no chromosomes at all. So in the case of Down syndrome, some of the cells that were made do not have the right number of chromosomes. And in this case, at chromosome 21, it actually has 3. So it's called a trisomy because it has 3 when you're only supposed to have 2. Okay, like I said, we'll get into genetic disorders. I just kind of want to talk about that, throw that in there. Okay, so I have a question for you. A laboratory assistant was told to watch the mitosis of a eukaryotic cell under the microscope and inform the lead scientist when the cell enters metaphase. So look at these pictures right here, and you're going to tell me which of these looks like metaphase. What should the assistant expect to look for to indicate that the cell is in metaphase? What was metaphase? I'll give you a hint. I'm just going to circle this letter. Well, metaphase means middle. So any chromosomes that are lined up in the middle of the cell. So right here, does it look like chromosomes are lined up in the middle of the cell? No, it looks like a big blob. How about this? Does it look like the chromosomes are lined up in the middle? No, it's a little bit less of a blob, but still pretty much a blob. How about this? Does this look like anything is lined up in the middle? Perhaps. Still looking a little blobbish, but it's looking a little bit more straight, so maybe. Let's look at the other options. How about here? Does this look like something is lined up in the middle? No, it looks like they're kind of already being pulled apart. Okay, how about this? Does it look like anything's lined up in the middle? No, the chromosomes are already forming 2 different nuclei. So, which one do we think is metaphase? C. Definitely C. Okay? So as you can see, like we looked at those cartoons, but this is what it looks like under an actual microscope. And obviously if we were in person, you guys would be able to look at the microscopes and whatnot, but unfortunately coronavirus prevents us from doing labs at the moment. Okay, last but not least, here is a cute little Kirby thing. It helps you with mitosis. So, yeah. This is interphase. This is going to be prophase when the cells are getting ready. Cells are getting ready. We have metaphase because they're in the middle. Anaphase because they're being pulled apart. Telophase, we have 2 new nuclei. And then cytokinesis, which is we have 2 totally different cells and whatnot. Okay, think of it like Kirby. That's Kirby. I was going to say Jigglypuff, but it's definitely Kirby. All right.